Thank you to Ataxia UK for inviting me to speak to you today. I'm Emma Davies and I'm a principal scientist at Helix. Today, I'm going to talk to you about Helix and what we are doing to find new treatments for rare diseases. So today, I'm first going to talk to you about the Rare Disease Challenge and Helix's mission. Then I will cover how Helix approaches drug discovery and our technology. Finally, I will share some of our preliminary unpublished preclinical data from our Friedreichs ataxia program. So I know this audience is fully aware of the Rare Disease Challenge, but for context, I'm going to talk through these facts and figures. There are over 7,000 known rare diseases, and only as few as 5% of these have a treatment. There are 400 million people globally that are living with a rare disease, and 50% of these are children. To put this into context, this is a greater number of people than the entire population of the USA, or roughly 1 in 17 people. So collectively, rare diseases are really not that rare. Helix was founded on a mission to serve this population. Our mission is simple. We want to achieve unprecedented impact for rare disease patients by pioneering the next generation of drug discovery. So who are we? Helix was founded in 2014 in Cambridge in the United Kingdom by Dr. Tim Williams and Dr. David Brown, the co-inventor of Viagra. We are nearly a 100-strong team of experts in technology, drug discovery, and clinical development. This screenshot captures some of the Helix team during one of our all-team meetings on Rare Disease Day this year. All our meetings are still virtual. So what is the Helix approach to drug discovery? Why is it different? So we are an artificial intelligence or AI company, and we use this to identify and progress novel rare disease treatments at scale. To do this, we combine our AI platform, HealNet, with a drug discovery expertise and patient insight to identify drugs that could be redeveloped, combined, or enhanced to treat rare diseases. So why are we different to the traditional model of drug discovery and clinical development? So the traditional model of drug discovery and clinical development of new medicines is very lengthy and expensive. To bring a new drug to market this way typically costs around $2 to $3 billion and takes around 12 to 14 years to develop, and unfortunately has a 95% failure rate. Our approach allows us to accelerate the speed of this process, increase the scale, and improve our chances of success. Many companies that develop drugs in the traditional way don't have the resources or risk tolerance to take drugs all the way for rare diseases, as the small patient population means any treatment a company develops will have to be extremely expensive in order to recoup the discovery and development costs. So this is a snapshot of our clinical pipeline at the moment. So the main therapeutic areas we are working on are rare neurological, oncological, renal, neurodegenerative, and muscular conditions, but we are constantly re-evaluating which conditions we work on. Our most advanced program is Fragile X syndrome, which has recently been approved to start clinical trials. So I'm going to talk to you a little bit more about our technology platform. So HealNet is our AI drug discovery platform. It integrates data from multiple sources, including biomedical research, patient group data, human domain expertise, and scientific literature to form the world's most detailed rare disease knowledge graph, which is represented graphically on this slide by this cluster-like structure at the center. This graph is then analyzed by our AI models to uncover novel connections between drugs, diseases, and other relevant medical information. HealNet then provides our team with a ranking of the most likely drugs and combinations that could impact a particular disease that we are working on. Our drug discovery scientists, including myself, then review these predictions. Some will seem obvious to us, but other predictions will be very novel. We then use our scientific and medical knowledge about the disease and the drugs to select the most likely drugs to treat the disease we are working on. Then these drugs are validated in preclinical models and progress towards the clinic. So now I'm going to talk to you a bit more about a program that we're working on in partnership with Ataxia UK. And this is our Friedreichs ataxia program, also known as FA. This is an autosomal recessive disease that is typically diagnosed by the onset of ataxia during puberty. Patients have progressive nervous system damage that leads to progressive ataxia, extreme fatigue, muscle weakness, impaired speech and swallowing, hearing loss, and scoliosis. 75% of FA patients also have cardiac hypertrophy, with heart failure being a leading cause of death in FA patients. Some patients will also develop diabetes and glucose intolerance. Friedreich's ataxia is a slow progressing disease with no approved therapies that treat the root cause of the disease. So what causes Friedreich's ataxia? There is a single causative gene in people with FA. This gene is called FXN and produces a protein called frataxin. In FA patients, there is an error in the genetic code for the FXN gene, whereby a large piece of DNA where the same 3 nucleotides, GAA, is repeated many times. This repetition of DNA is referred to as expansion. And this expansion means the message of the FXN gene is not transcribed properly. And this leads to loss of the protein product frataxin in the cells of FA patients, which is exemplified by this graph. So what does loss of this protein frataxin mean to the cell? The frataxin protein is specifically found in the organelles of cells known as mitochondria. These are energy-producing structures that are regarded to be the powerhouse of the cell. The function of frataxin is currently unknown, but it is thought to play a role in the storage of iron and the formation of iron-sulfur clusters in the mitochondria, which are critical for its function. Excess iron is observed in FA cells, and this leads to overall mitochondrial dysfunction, a depletion in energy production, and ultimately leads to degeneration of the spinal cord and peripheral nerves and the cerebellum, which causes the symptoms of Friedreich's ataxia. So we used our HealNet platform to predict drugs that might be efficacious in FA, with a focus on drugs that might alter the mitochondrial function and treat the root cause of disease. These predictions were reviewed, and a short list of drugs were tested in vitro in a cell model or fibroblast model of FA. We measured the respiration or oxygen consumption of the cells, which is an indicator of mitochondrial activity. A specific measure of respiration called excess reserve capacity was calculated. And this, as you can see on this graph on the right, you can see that the excess reserve capacity of FA cells, which is represented by this blue bar, is much lower than the non-FA control cells, which is represented by the red bar. The green bars represent excess reserve capacity for FA cells that have been treated with our 3 Helix compounds as single agents. You can see that all 3 drugs increase the excess reserve capacity to values that are greater or similar to the control non-FA cells. So to summarize, I have told you about Helix's mission and how we are using AI and machine learning to take existing drugs and redevelop, enhance and combine drugs for rare disease patients. Using our technology, we have identified 3 compounds that restore mitochondrial function in a cell model of Friedreich's ataxia. These compounds are all approved drugs used to treat common diseases. The next steps will be to understand if these compounds will work in a whole body animal model of Friedreich's ataxia and prevent the symptoms of Friedreich's ataxia before testing them in FA patients in randomized controlled clinical trials. So I'd like to take the chance to thank you for listening to me today. Specifically, I would like to thank the Helix Friedreich's ataxia project team, our collaborators at Nottingham University that carried out the preclinical work that I shared with you. Thank you to Ataxia UK for inviting me to speak to you and collaborating with us on this project. And finally, and most importantly, thank you to all the rare disease patients that help us with our understanding of disease and participate in research studies. Thank you.